Hi everyone, today let me talk about the different soil organisms and different microbiological processes. Before we move on, let us first define what is soil microbiology. Soil microbiology is the study of organisms in soil, their functions, and how they affect the soil properties in relation to plant growth. So in this video, we will tackle some important organisms living in the soil and their relationship with other soil properties such as the physical and chemical, and we will relate those properties in plant growth. Another definition of soil microbiology is that it is a scientific discipline that is concerned with the study of all biological aspects of life that is existing in the soil. Now let us define what is a soil organism. Soil organism is any organism inhabiting the soil during part or all of its life. It ranges in size from microscopic cells that digest decaying organic material to small mammals that live primarily on other soil organisms. Soil organisms play an important role in maintaining the fertility, the structure, drainage, aeration, and other properties of the soil. Soil organisms play a very crucial role in the soil. They decompose organic residues. They are the ones recycling the nutrients from those organic residues. And they can also enhance soil structure. They clear away and degrade organic debris such as dead plants, animals, and dung, and use it as source of food and nutrients. And in the process, they release chemical elements into the soil solution so that other living organisms can reuse it, such as the plants. There are two kinds of, or two general classifications of soil organisms based on the size. And these are the following: micro and macroorganism. So microorganisms are the smallest of the soil animals, ranging from 20 to 200 micrometers in length or less than 0.01 micrometers in diameter. They are extremely abundant and diverse and can decompose all existing natural material in the soil. This includes bacteria, fungi, protozoa, nematodes, algae, actinomycetes, and viruses. Microorganisms are those organisms that cannot be seen with the naked eye. You need a microscope to be able to see them. They exist more in the topsoil, where food sources are plentiful, than in subsoil, and they are especially abundant in the area immediately next to the plant roots. So this area is also called the rhizosphere, where slough of cells and chemicals released by roots provide food sources. Microorganisms are the primary decomposers of organic matter. They provide nitrogen through fixation to help growing plants, and also they detoxify harmful chemicals. They can also suppress disease organisms and produce products that might stimulate plant growth. And also they have direct importance to humans. They can be source of antibiotics. They are the source of most of the antibiotic or medicines we use to fight diseases. So as I've said earlier, microorganisms include the following: bacteria, fungi, protozoa, nematodes, algae, actinomycetes, and viruses. So let's take a look in all those. So the first one is the bacteria. Bacteria are small, single-celled organisms that make up the single most abundant type of microbe. And these organisms can withstand very wide range of conditions that they live in, from the Arctic wastelands to the steaming waters of volcanic hot springs. In soils, they multiply rapidly under the proper conditions. Well, in fact, in a teaspoon of soil, about 5 to 7 grams of soil, they are typically thousands in terms of volume. They are decomposers, they eat dead plant materials and organic waste, and by doing this, they can release nutrients that other organisms could not access. And also, they can transform nutrients from inaccessible to usable forms. In other words, they can mineralize unavailable soil nutrients for plant growth. So, they can benefit the plant by increasing the nutrient availability from the soil. Many of bacteria this can dissolve um, insoluble phosphorus, making it more available for plants to use. They are also very helpful in providing nitrogen to plants, which they need in large amounts but is often deficient in agricultural soils. Examples of those are what we call the nitrogen fixing bacteria, the nitrifiers, and those processes will be discussed towards the end of this video. Next we have is fungi. Fungi play a critical role in the decomposition of dead and or living organic material. They are helpful but could also be harmful to soil organisms. They are helpful because they can break down nutrients that other organisms cannot. And also, they can release them into the soil and other organisms get used to them. Fungi can attach themselves to plant roots, and most plants grow much better when this happens. An example of this is the beneficial relationship called mycorrhizae, in which the fungi help the plant by giving it needed nutrients, and the fungi get carbohydrates from the plant, the same food that plants give to humans. On the other hand, fungi can get food by being parasites and attaching themselves to plants or other organisms. Fungi can be grouped according to their functions performed in the soil. They can be the decomposers, they can be parasites, or they can be mutualists. Decomposers are also called saprophytic or saprophytic fungi. They convert dead organic material into fungal biomass, carbon dioxide, and small molecules such as organic acids. Mutualists, the common example is the mycorrhizal fungi. They colonize the plant roots and exchange for carbon from the plant. Mycorrhizal fungi help to make phosphorus soluble and bring soil nutrients such as the phosphorus, nitrogen, and other micronutrients and perhaps water to the plant. The hyphae, in this figure you can see the hyphae of the mycorrhiza. The hyphae of this fungi help develop and stabilize larger soil aggregates by secreting a sticky gel that glues mineral and organic particles together, meaning it can also improve soil aggregation and soil structure. There are two kinds of mycorrhizal fungi. We have the ecto and endomycorrhizae. Ectomycorrhizae grow on the surface layers of the roots and are commonly associated with trees, while endomycorrhizae grow within the root cells and which are commonly associated with grasses, root crops, vegetables, and shrubs. In other words, endomycorrhizae are those mycorrhiza that grows in plants with relatively finer or smaller roots in diameter, while ectomycorrhizae grow commonly in trees wherein the roots of trees are actually really large compared to grasses, vegetables, and shrubs. And the third one is, or are the parasites. Fungi could be a pathogen or parasites. They can impair production or death. They can cause death when they colonize the organisms. Fungi are less sensitive to acid soil conditions than bacteria. They love acid soils, but they cannot function without oxygen. Low soil disturbance resulting from reduced tillage systems tends to promote organic residue accumulation at and near the surface. Tends to promote fungal growth, as happens in many natural undisturbed ecosystems. Next to fungi is protozoa. Protozoa are colorless, single-celled animal-like organisms. They are larger than bacteria, varying from a few microns to a few millimeters. Their population in arable soil ranges from 10,000 to 100,000 per gram of soil, and they are abundant in surface soil. They can also withstand adverse soil conditions as they are characterized by a protected dormant stage in their life cycle. So in this figure is an example of protozoa, Paramecium bosaria, a ciliate which derives some of its nutrients from algal endosymbionts in the genus Chlorella. Protozoa are requiring bacteria to eat and water for movement. They also feed on each other and can reduce bacterial diseases by consuming large number of pathogenic bacteria. And the regulation of bacterial population through grazing can release excess nitrogen in the form of ammonium. Therefore, they can provide nitrogen for plant growth, as protozoa generally occupy soil zones within the rhizosphere adjacent to root structures. Another soil microorganisms are the nematodes. Nematodes are microscopic worms, typically measures 50 microns in diameter and 1 millimeter in length. They live in water surrounding root zones, and they are the final microbiological life form responsible for living soil. Excessive irrigation favors some parasitic omnivore nematodes, which may greatly reduce crop production. And nematodes also consume fungi, bacteria, protozoa, and each other, returning nutrients to the soil and maintaining balanced ecosystem within the soil. The free-living nematodes can be divided into three broad groups based on their diet. We have the bacterial feeders, the fungal feeders, and predatory nematodes. Of course, bacterial feeders are those which consume bacteria. Fungal feeders are those consume fungi by puncturing the cell walls and sucking out the internal contents. And the last one, the predatory nematodes eat all types of nematodes and protozoa. They eat smaller organisms whole or attach themselves to the cuticle of larger nematodes, scraping it until the prey's um, internals can be extracted. Like protozoa, nematodes are important in mineralizing or releasing nutrients in plant available forms. When nematodes eat bacteria or fungi, ammonium is released because bacteria and fungi contain much more nitrogen than the nematodes require. They may also be useful indicators of soil quality because of their tremendous diversity and their participation in many functions at different levels in the soil food web. Next we have is algae. Algae are present in most of the soils where moisture and sunlight are available. Their number in the soil usually range from 100 to 10,000 per gram of soil, and they are capable of photosynthesis just like the plants. They can obtain carbon dioxide from the atmosphere and energy from sunlight and then synthesize their own food, which they can use for energy and to help build other molecules they need. They are found in abundance in the flooded soils of swamps and rice paddies, and they can be found on the surface of poorly drained soils and in wet depressions. They may also occur in relatively dry soils, and they form mutually beneficial relationships with other organisms. Example of this are the lichens. Lichens found in rocks. Lichens are the association between a fungus and an algae. The major roles and functions of algae are they play an important role in the maintenance of soil fertility, especially in tropical soils. They can also add organic matter to soil when they die, and thus increasing the amount of organic carbon in soil. They could also act as cementing agent by binding soil particles and thereby reducing and preventing soil erosion. In addition, algae can help increase the water retention capacity of the soil for longer periods of time. They can liberate large quantities of oxygen in the soil environment through the process of photosynthesis and thus facilitating submerged aeration. They can also help in checking the loss of nitrates through leaching and drainage, especially in uncropped soils. And they can help in the weathering of rocks and building up of soil structure. The next organism are the what we call the actinomycetes. Actinomycetes are like both bacteria and fungi and have characteristics linking them to both groups. They are often believed to be the missing evolutionary link between bacteria and fungi, but they have many more characteristics in common with bacteria than they do with fungi. Actinomycetes give the soil its characteristic smell. They have also been the source of several significant therapeutic medicines and are also necessary for the breakdown of certain components in organic matter. The next organism we have is the virus or the viruses. Soil viruses are of great importance as they may influence the ecology of soil biological communities through both an ability to transfer genes from host to host and as a potential cause of microbial mortality. Soils probably harbor many novel viral species that together may represent a large reservoir of genetic diversity. Some researchers believe that investigating this largely unexplored diversity of soil viruses has the potential to transform our understanding of the role of viruses in global ecosystem processes and the evolution of microbial life itself. So that's all for the soil microorganisms. Now we move to the next level. We will discuss some examples of the macroorganisms. And to define macroorganisms are organisms that are visible to the naked eye, which are generally greater than 2 millimeters in diameter. This includes earthworms and arthropods such as the ants, termites, millipedes, centipedes, caterpillars, and so on. Macroorganisms are called ecosystem engineers because they burrow and are important in mixing the soil, and this process is known as bioturbation. Most soil macroorganisms occur in the top 30 centimeters of soil, although some also occur at depth. These organisms may move to lower soil layers when conditions at the surface are harsh. Now let's have a brief discussion about arthropods. Arthropods are the primary macroorganisms responsible for maintaining the health of our living soils. They constitute a population of predator or prey organisms which facilitate nutrient release, recycling, and utilization. They are grouped as shredders, predators, herbivores, and fungal feeders. And they all occupy critical roles with respect to maintaining healthy living and productive soils. Larger arthropods live on the soil surface and shred organic debris and each other into small parts, aiding the decomposition and incorporation of organic matter into the soil. Examples of arthropods are the millipedes, the sobags, the earwigs, and some beetles. Other arthropods such as the spiders and pseudoscorpions predate each other and the other shredders. The results of all of this frenetic interaction are copious streams of recycled organic material and body parts circulating throughout the soil ecosystem. Another example of arthropod is the springtail. Springtails, along with nematodes and mites, share the numerical dominance among soil invertebrates. They feed principally on fungi, although they also eat nematodes and small bits of organic detritus. They are among the most important soil organisms and are a major controlling factor of fungi populations. We also have the millipedes. Millipedes are vegetarians that are helpful in breaking down organic matter. Millipedes are a worm-like body, have a worm-like body, and adults can grow to a length of 1 to 2 inches. And also we have the flies. Flies, including the house flies, play an important part in the recycling and breaking down of all types of organic debris. Adults can feed on almost any kind of organic material. We also have the sawbugs and peelbugs, or the isopods. They are small, flat-bodied, flat decomposers that closely resemble each other. The way to tell them apart is by the fact that only peelbugs are able to roll up into a ball. Like other crustaceans, sawbugs breathe through gills and require moist environment. They feed on rotting wood and highly durable leaf tissues. We also have the ants. Ants in an open air compost pile may also be an indicator that the pile is too dry. Ants may even benefit the composting process by bringing fungi and other organisms into their nest. The work of ants can also make compost richer in phosphorus and potassium by moving minerals from one place to another. So that's the role of ants. We also have the earwigs. Earwigs are among the most readily recognized and most commonly complained about insect pests in gardens. Earwigs forage at night, eating the eggs, young, and adults of small organisms such as insects, mites, and nematodes, as well as algae, fungi, and other plant tissues. And we also have the common earthworm. Earthworm alter and mix soil structure to better facilitate water movement and water holding capacity. They bury and shred surface organic material. They can provide channels for root growth and development, can maintain nutrient dynamics, facilitate plant growth, and stimulate microbial activity. Earthworms pass organic matter through their bodies, grinding it with the help of tiny stones in their gizzard. The material passes out of the worm's body in the form of worm castings, which are the richest and finest quality of all humus material. That is why we have what we call the vermicomposting. The fresh castings are markedly higher in bacteria organic material and available nitrogen, calcium, magnesium, phosphorus, and potassium than soil itself. And as worms process organic materials, they coat the materials with a mucus film that binds small particles into stable aggregates, therefore protecting the nutrients from being lost out by rain. So those are the examples of macroorganisms. Now we are moving on with another topic, which is all about microbiological processes. So these are some of the soil microbiological processes. We will not discuss all of the microbiological processes happening in the soil, but these four are the most commonly discussed or common ones in terms of soil science. So you have the microbial transformations of nitrogen, the inorganic P-solubilization, microbial transformation of sulfur, and iron precipitation by soil bacteria. So let's discuss this for one by one. In terms of microbial transformations of nitrogen, the first one on our list is mineralization. Mineralization is the conversion of organic nitrogen to inorganic nitrogen, and this renders nitrogen available for plant use because plant can only uptake the inorganic forms of nitrogen and not the organic forms. Another microbial transformation is what they call the immobilization. This is the opposite of mineralization because immobilization is the conversion of inorganic N to organic N, and this renders nitrogen unavailable for plant use. And immobilization happens when available nitrogen is used up by the microorganisms and incorporated it in their bodies. Another microbial transformation is what we call the nitrification or nitrification. It is the biological formation of nitrites or nitrates from compounds containing reduced nitrogen. And the most common initial substrate is ammonium, and the final product is nitrate. There are two separate and distinct steps in this process. First is the initial oxidation of ammonium to nitrite, that is NO2 minus. Second is further oxidation of nitrite to nitrate, that is NO3 minus. The production of nitrate is related to soil and solution pH values. The optimum pH values vary from 6.6 to 8, and rate decreases below 6 and becomes negligible below pH 4.5. The microorganisms responsible for nitrification process is nitrosomonas. Nitrosomonas are chemoautotropic, gram-negative, non-spore-forming, ellipsoidal or short rods, ammonium-oxidizing bacteria, and they are responsible for the oxidation of ammonium to nitrite or NO2 minus. And the one that is responsible for oxidation of nitrite to nitrate is what we call the nitrobacter. Nitrobacter are also chemoautotropic, also non-negative. They are also short rods bacteria. Nitrate can be lost through leaching through the nitrification and in leaching, particularly in sandy soils. Why is that so? Because sandy soils have a lot of macropores. That is under excessive rainfall or heavy rainfall or under excessive irrigation. Excess nitrate, leached from soil, often ends up in groundwater, lakes, streams, and this can lead to water pollution such as eutrophication or the excess growth of plant and algae, and some health problems in infants and animals, or what they call methemoglobinemia. As mentioned in the previous slide, another microbial transformation of nitrogen is what they call denitrification. It is a biochemical reduction of nitrate to gaseous nitrogen by facultative anaerobic soil organisms. The denitrificants reduce the nitrate to nitrite, then to gaseous nitrogen forms, which is nitrous oxide, or it can be elemental nitrogen that are commonly lost to the atmosphere. The nitrification is all aerobic, but nitrate is used as the electron acceptor in the absence of oxygen. The nitrificants or the denitrifying organisms require a very low oxygen concentration of less than 10% and organic carbon for energy. The microorganisms involved in denitrification are Pseudomonas, Achromobacter, Bacillus, and Micrococcus. Denitrification is a major avenue of loss of nitrogen in paddy soils, and it contributes to the low efficiency of applied nitrogen fertilizer because nitrogen fertilizer can be easily denitrified or volatilized. Nitrate in the thin aerobic surface of paddy soils is leached down to the reduced subsoil. We also have what we call ammonification. The process of ammonification is the result of the breakdown of organic matter such as dead animals in plants or waste materials like excrement. This breakdown is accomplished by microorganisms which utilize dead organic material for energy and then produce ammonia and related compounds as byproduct of their metabolism. We also have what we call symbiotic biological nitrogen fixation. So this process renders nitrogen available to plants. It occurs commonly in legumes in the presence of rhizobia. Rhizobia is a nitrogen-fixing bacteria which are aerobic, gram-negative, non-spore-forming, and typically motile with simple nutritional requirement. Rhizobia enters the root hair of the plants, causing the formation of root nodules. The bacteria in root nodules trap atmospheric nitrogen, which is transformed to ammonia, and this will combine to organic compounds to form amino acids and proteins. In return, the plant supplies the carbohydrates and energy for the bacteria's metabolism. And then when the legumes are plowed into the soil, the fixed nitrogen is added into the soil. We also have what we call non-symbiotic nitrogen fixation. It is the conversion of atmospheric nitrogen amounting to 20 to 100 kilograms nitrogen per hectare per year. That is done by microorganisms without an associated plant host. And the organisms involved are the free-living bacteria such as Azotobacter, Begioringia, and Clostridium pastorianum, and some algae and other species. Azotobacters are strict aerobes. They are mesophiles with an optimum temperature of 30 degrees Celsius. They are gram-negative and their shape is large cocci. The members of genus Clostridium are anaerobes. They are gram-positive rods which are found in soils with pH 5 and are still capable of growth at pH 9. Other organisms involved in non-symbiotic nitrogen fixation are the Clostridia and the blue-green algae. Clostridia proliferate when organic matter is added and they are often numerous around the plant roots. BGA or blue-green algae are believed to maintain the fertility of lowland rice. And the following is a list of commonly uh, common BGAs in the Philippines, from Anabena to Tolipotrix. So we are finished with the microbial nitrogen transformations. Now let's look on the inorganic P solubilization. So the following are the genera of bacteria capable of solubilizing calcium phosphates. We have Pseudomonas, Mycobacterium, Bacillus, and Micrococcus. While on the list, another list of microorganisms that is for fungi, capable also of solubilizing calcium phosphate, we have Penicillium, Fusarium, and Aspergillus. Inorganic P solubilization is a microbiological means by which inorganic P is solubilized. It involves the production of organic acids, nitric acid or sulfuric acid production, and flooding, resulting in the reduction of iron in insoluble ferric phosphates. This can involve also mycorrhizal association. As discussed earlier in this video, there are two general types of mycorrhizal association, the ectotropic and the endomycorrhiza. Ectotropic involves the fungus forming a mantle around root exteriors, and then the hyphae of the fungus enters into spaces between the plant cells. And these are commonly involved in pine and eucalyptus. For the endomycorrhiza, the fungus penetrates the cells of the plants. For examples, we have the orchids, the coffee, fruit trees, rice, and corn. We are finished now with the microbial transformations of nitrogen and phosphorus. Now we move to the microbial transformations of sulfur. This involve the conversion of organic sulfur to inorganic sulfur. Because the major sulfur in soil is in the organic fraction with low concentration of sulfate. That is the form available for plants. This involves the following transformation: the decomposition of organic sulfur compound, the microbial assimilation or immobilization of sulfur and their incorporation into microbial cells, the oxidation of inorganic compounds such as sulfides, thiosulfates, and elemental sulfur, making it available for plant uptake, and the reduction of sulfate and other anions to sulfide. In anaerobic conditions, sulfate availability may be limited in the soil. The sulfate-reducing bacteria, predominantly of the genus Desulfo, Vibrio, and Desulfo tomaculum, use sulfate as the hydrogen acceptor in their energy-yielding metabolism that can reduce sulfate to sulfide, making it not available for plant uptake. While in aerated environments, the combined sulfur is ultimately metabolized to sulfate. The members of the genus Thiobacillus are capable of oxidizing elemental sulfur to sulfate, making it available for plant uptake. They are gram-negative and non-sporulating rods, which are predominantly aerobes. The oxidation of elemental sulfur leads to the formation of enormous amounts of sulfuric acid, of which decreases soil pH. So, microbial transformation of um, elemental sulfur to sulfate can acidify the soil. So, that's it. We have discussed microbial transformations of nitrogen, phosphorus, and sulfur. Now, for the last common um, microbial process in soil, we have iron precipitation by soil bacteria. So this process is brought about by a group of bacteria sometimes termed as the iron bacteria. And these transformations involve ferrous oxidation from Fe2 to Fe3 plus, also iron reduction, and iron precipitation of from organic salt. So that's it. So those are the microbial processes that are happening on the soil. I hope you learned from this video. Please don't forget to like and subscribe.